really understanding how we tick is really what Nick King, Associate Director of the Translational and Safety Sciences program, is going to talk to us about. Nick has been with the Critical Path Institute for over 10 years. And as I said, Nick is going to go into a little bit of depth of around what we've evolved into and how we actually work. Nick, floor is yours.
Great, thank you very much, John Michael, and thank you to our keynote speakers, as well as Laura Hawkins for organizing today's meeting. And all right, so I'm going to speak a little bit about how PSTC operates as a public-private partnership, and how how the members utilize this space as a pre-competitive area to work collaboratively on projects that benefit everyone involved in drug development, from company to patient. And so I'm going to go through a little bit of our mission and structure that you've heard quite a bit already today, and then could walk us through what a project looks like for PSTC. All right, as everyone has heard a few times already, we were formed in 2006 in March, and our goal is to share and validate innovative safety testing methods. We do this in a pre-competitive space where member companies share data from studies that they've conducted across the membership. We collect all of that data and then analyze it as a single data set, with the primary goal within PSTC to obtain regulatory qualification or endorsement of novel translational, using both non-clinical and clinical studies, safety biomarkers for use in drug development. We have, the consortium is comprised currently of 13 member companies and partnerships with several different agencies and research groups globally, including the EMA, US FDA, the Japanese PMDA, as well as FNIH, the Innovative Medicines Initiative in Europe, SRI International, as John Marsalis spoke to earlier, as well as the Japanese NIHS. Now, our approach has been to have a translational perspective on advancing fluid-based safety biomarkers. And so we do this by first conducting experiments in non-clinical species, including rats, canine, and non-human primates, and comparing the performance of the fluid safety biomarkers to histopathology in those species, and looking at how well the biomarkers determine injury, the specific injury in those studies when toxicants are given. And then from those biomarkers that perform well and we're confident at detecting specific injury, we advance those into clinical studies, looking at how they perform in the clinical space and at detecting injury in studies where there may be some level of injury due to the drug given to a patient. And in the case of humans, we don't have the option to utilize histopathology frequently, and so we're dependent upon fluid-based safety biomarkers. Currently, we have a focus in 6 different tissue areas, and that includes the kidney, skeletal muscle, liver, the vascular system, the testicular toxicity, as well as pancreatic injury. And you'll see in each of these, they have different issues around sensitivity and specificity to detect injury in each of those tissues. And so those are some of the limitations that we're looking to improve upon through the collaboration of our consortium. And so we are organized with an advisory committee that's comprised of a representative from each of our member companies, as well as the c-path staff, executive director, and a liaison from the FDA. And then as well, the co-chairs to each working group, and our working groups are organized around the specific tissue or organ injury. And that is where the research is conducted. So the advisory committee provides the strategic direction, and the working groups execute on that strategy. And so I just want to kind of dive a little bit deeper into how we do this in partnership with our members, and how our c-path staff, as well as member company scientists, contribute to the work that we produce. So as I mentioned, the advisory committee determines a strategic direction for the consortium in partnership with our executive director, project managers, and project coordinators. And then as we look at a working group, the working groups, looking at that strategic direction, develop and oversee organ or tissue specific objectives. And there we bring in scientists with the specific expertise to a liver group, or to a working group. In this case, I have our hepatotoxicity or liver injury working group as an example. And again, now on the c-path side, we have the executive director providing scientific input, our project managers keeping all of us on tasks, as well as the project coordinators helping to keep us on task and keep the meetings moving forward. We add in the data managers who collect the data from each of the contributed studies and map that into a common template and merge it into a final data set that we can analyze. And then as we move into a more specific, we look at executing on these individual projects to address the objectives of the working group. And so here I have two examples within the hepatotoxicity working group. The first is the glutamate dehydrogenase qualification as a safety biomarker to detect drug-induced liver injury. And then the other is our bile acid team. And so each of those have specific research objectives and plans that they've developed and are executing on. And again, this is where we bring in our scientific expertise from the member companies, as well as the operational and logistics support from our c-path project managers, project coordinators, and data managers, as well as scientific leadership from our input from our executive director. And to kind of further move into how this works within an individual project, I'm going to use our glutamate dehydrogenase qualification, or GLDH, as I'll refer to it. So, you know, one of the pieces that we've seen is liver injury is a major concern in drug development. It leads to the cessation of many drug development programs. And one piece is being able to specifically and sensitively detect liver injury. And specifically as well, in the case of drug trials where there may be elevated serum transaminases due to causes external to the liver, such as in muscle injury or degeneration in patients such as those with muscular dystrophy. And so in this case, GLDH is a liver enzyme that is primarily expressed in the liver with trace amounts in a few other tissue, but the serum release can be primarily tracked back to the liver. And so this group has been working for a few years now, and over the next few slides, I'll walk us through what they have conducted. And before we move into that, I just want to touch on how we bring into the working group and consortium the data. So we have a consortium study data that is contributed from our member companies. That's both non-clinical as well as clinical studies. PSTC also did conduct several years ago a normal healthy volunteer study in order to obtain baseline and reference range data for our clinical qualifications. Our Data Collaboration Center data managers then bring that data in, they curate and standardize all of it, and then load it into an integrated database from which we can output data sets to be analyzed by our statisticians. And then based on the results of that statistical analysis, have an actionable safety biomarker or drug development tool that we pursue regulatory review and endorsement currently at 3 regulatory agencies. That's the EMA, the PMDA, and the US FDA. And then finally, once we obtain those regulatory endorsement, those are posted publicly, the tools, these safety biomarkers, and how they may be utilized with a specific context of use. It's made publicly available. We post those on our website, and then they're also on the health authorities' websites as well. And now, within the case of the GLDH qualification, we took this, our translational approach, in several steps. And so the first was looking at non-clinical studies. And the objective here was to establish the sensitivity and specificity of GLDH to detect drug-induced liver injury in a rat model. And to achieve this, member companies contributed over 30 studies with more than 20 different toxicants that were either specific to the liver or to other organs. So for specific to the liver, to look at sensitivity to detect liver injury, and then in data sets where the toxicant was specific to another organ to determine the specificity of GLDH to only detect liver injury. And once we saw a very positive correlation between the changes in GLDH and the level of histopathologically determined liver injury in these rats, we advanced GLDH to look at some exploratory studies in humans. And there, our objective was to determine a reference range and thresholds of concern for drug-induced liver injury. And here, we have several studies that were contributed by members, as well as the PSTC conducted normal healthy volunteer study. And once this information was incorporated, analyzed, and summarized, we submitted a qualification plan to the FDA and to the EMA for review and acceptance of what we proposed as a set of confirmatory clinical studies. And once we had alignment on those studies, we executed on the confirmatory clinical studies where we were testing the sensitivity of the reference range that we established, as well as the cut points or thresholds of concern for DILI. And then we also tested the specificity of GLDH in studies that had patients with pancreatic injury, kidney injury, as well as gastrointestinal injury. And so, again, these studies were contributed by PSTC members. And as well, in a collaborative spirit, we have been working with c-path's Duchenne Regulatory Science Consortium, as they have a specific interest in having a biomarker that can detect liver injury in patients with Duchenne's muscular dystrophy, where one of the normal biomarkers for detecting liver injury is extremely elevated at baseline for those boys, and so is not available to detect liver injury as sensitively as desired. So GLDH is a perfect case for applying that in a specific disease population. And so, in summary, I just would like to come back and say that, PSTC, we pursue a translational approach that to advance regulatory acceptance of promising novel safety biomarkers. In this case today, I spoke to glutamate dehydrogenase as a safety biomarker to detect drug-induced liver injury. We have early work that incorporates input from industry, academic, and regulatory agency research for these novel safety biomarkers. And then, shared data from the GLDH qualification project has already led to 2 publications, as well as a letter of support from the EMA that this is a biomarker of interest to pursue further. And so, finally, advancing safety biomarker qualification projects work when several stakeholders are actively engaged and involved in contributing data, and that's the key to the success of PSTC. So I'd like to thank all of our members, as well as our c-path staff, for the contributions that you've made. And finally, I would like to acknowledge the c-path staff I work with on a daily basis. You've all been key and crucial for all of the work that we conduct every day, as well as putting together this wonderful workshop. So thank you very much.